So let's get started, even though some other people may join soon. All right. So it's my great honor and pleasure to introduce our speaker today. I used to think that the academic world is small, but now I think the world is small. And let me tell you why. Dr. Meng and I were both born in the same province in China, Shandong province. And actually he went to college not very far from the college I went to, even though, of course, he went to college earlier than I did. And now, decades later, both of us are working and living in state of Virginia, in US. So that's what I really meant by the world is small, or at least it seems to be so small. But of course, Dr. Meng is much more accomplished researcher and scientist than I am. I cannot even think of achieving the same level as he has already done now. So Dr. Meng is currently a university distinguished professor and the director of the Virginia Tech Center for Emerging Zoonotic and Arthropod-borne Pathogens. And Dr. Meng is also an elected member of the National Academy of Sciences and an elected fellow of the National Academy of Inventors. And Dr. Meng, as I just mentioned, he received his medical degree in 1985 from Binzhou Medical University. And he received his Master of Science degree in microbiology and immunology in 1988 from Wuhan University School of Medicine, so which was formerly called Hubei Medical University. And he received his PhD in immunology in 1995 from Iowa State University. And from 1995 to 1999, Dr. Meng worked in the lab of infectious disease at NIAID at AIH. First as a John Forkey visiting scientist, and later as a senior staff fellow. And in 1999, Dr. Meng joined the faculty at Virginia Tech, where he rose from assistant professor all the way to full professor, and is currently a university distinguished professor, as I mentioned earlier. So this is a really very preeminent lifetime title, and the highest effective recognition that could ever be bestowed by Virginia Tech Board of Visitors. So Dr. Meng's research, as we will talk about today, focuses on emerging and zoonotic viruses of veterinary and human public health significance, right? Including hepatitis E virus and coronavirus, such as SARS-CoV-1 and SARS-CoV-2, as we were just chatting about before we get started now. So Dr. Meng's research group discovered the Swan Hepatitis E virus from pigs, and even hepatitis E virus from chickens, and demonstrate their ability to infect across species barriers, which led to a paradigm shift, recognition of human hepatitis E as a zoonotic disease, right? Which I think is a very important milestone in the research of hepatitis E. So Dr. Meng's group had also been studying the molecular mechanism of virus replication and pathogenesis with emphasis on vaccine development for several important emerging viruses. Actually, he has been awarded 22 US patents, and with 17 still pending on virus vaccines against important emerging virus diseases, which again is a very important achievement. Dr. Meng had published more than 345 peer-reviewed scientific papers, reviews, and book chapters, a number I cannot even think of now. Dr. Meng is also ranked in the top 1% of highly cited scientists in the field of microbiology from 1997 to 2007. And his publication has been cited for more than 30,000 135 times. He had a H index very high, so high of 90, and I-10 index of 269, both of which are really very high, indicating of his great impact to the field. So he currently served as editor-in-chief or editor for 5 international journals, including PNAS, the Proceedings of National Academy of Science, MBio, Virus Research, Veterinary Microbiology, and Animal Health Research Review. And again, I mentioned that already Dr. Meng is the inventor with 22 awarded and 17 pending US patents on vaccines. And 2 of his vaccines actually have been licensed for commercial purpose, and which is currently available on the global market. So Dr. Meng has also served in many leadership positions at both national and international levels, including as the chair of the Summit on Emerging Infections and Preparedness at the National Academy of Sciences in 2017, which actually was hosted by the US Senator Mark Warner from our state of Virginia. And he was also co-chair of the 2016 American Society for Virology Annual Meetings Local Planning Committee. And he was the chair of the USDA NC 229 committee and a member of the board of directors of Virginia Academy of Sciences, Medicine, and Engineering, and also a member of the board of directors of Virginia Tech Intellectual Properties Company. And he was also he is also a chair of the NIH Special Emphasis Panel, IDMB, and the chair of the International PRS Symposium, as well as some other leadership positions. Right? He has received more than 30 prestigious awards and honors. For example, he received the inaugural Lorraine Hoffman Alumni Award from our state university, where he received his PhD. And he was also the recipient of the 2017 Outstanding Faculty Award by SHEV, the State Council of Higher Education for Virginia, so which is the highest honor bestowed to a faculty member at Virginia's both public and private college and universities, which again is a great honor and achievement. And he was also the honorary diplomat of the American College of Veterinary Microbiologists. And he received the Pfizer Award for Research Excellence in both 2001 and 2007. And not to mention, actually, this, I mean, as indicator of his great scientific accomplishment that have been recognized by the field, he was elected to the American Academy of Microbiology in 2012 and inducted into the National Academy of Inventors in 2015 and elected to the National Academy of Sciences in 2016. Again, all of them are great honors. So without further ado, let me give the floor to Dr. Meng, who will talk about some of his research that he has been conducting for a long time on hepatitis E. Right? So let's give our warm welcome to Dr. Meng. If you can clap.
Well, thank you, Dr. Xie, and for that really nice introduction. And good afternoon, everyone. It is a pleasure to speak here today. And what I'm trying to do here today is to discuss with you about a very important human pathogen, the hepatitis E virus. Particularly, I'm going to focus on the expanding host range and cross-species zoonotic infection by these important human pathogens. Now, as some of you probably know, that hepatitis E virus, or HEV, is one of the many viruses that cause hepatitis in humans. And if you look at these slides, you can see, and these are some of those well-known and well-characterized hepatitis viruses. The hepatitis A virus, or HAV, which causes an enterically transmitted acute viral hepatitis. and the hepatitis B virus or HBV. And let me try to find a pointer. There we go. The HBV that cause chronic hepatitis and also liver cancer. The hepatitis C virus or related viruses, and this is a group of viruses that also cause chronic hepatitis and that can lead to liver cirrhosis and liver cancers. The hepatitis D virus or HDV, which typically found during co-infection with the hepatitis B virus. And of course, the hepatitis E virus or HEV, which we're going to talk about here today. Now, the virus belong to the family of Hepiviridae. So this is a group of a single-stranded positive sense RNA viruses. The genome is kind of a mid-size, but 7.2 kb in size. Morphologically, if you look at this 2EM picture, the hepatitis C virus is in a spherical, small virus particles, and it's only about 30 to 35 nanometers in size. So the disease caused by the hepatitis C virus is called hepatitis E, and clinically, you really cannot distinguish the symptoms of acute hepatitis E from other types of acute hepatitis. And they are very similar. The patient would have jaundice, abdominal pain, nausea, and fever. So it's a typical acute viral hepatitis. The incubation period for the hepatitis C typically range from about 2 weeks to up to 2 months. And one of the unique features associated with the hepatitis C virus infection is the relatively high mortality that we see during pregnancy, which can be up to 25 to 30% in some infected pregnant women. The virus is transmitted fecal-orally, but also recent studies have shown that it can be transmitted through the zoonotic route, which is the focus of today's talk. And so this is really an important public health problem in many developing countries, especially in Asia and in Africa. But hepatitis C also caused sporadic cluster cases in industrialized countries, including the United States. More recently, the chronic hepatitis C virus infection has become really important clinical problems, especially in those immunocompromised individuals, such as organ transplant recipient. And also, HEV has been linked to a number of extrahepatic clinical manifestations, especially some of the neurological diseases, such as Guillain-Barré syndromes, neurological amyloidopathy, and also cause kidney disease. Let's see if I somehow, it does not allow me to advance the slides. Yeah, let's see here, what's going on? Let me do it this way. Here we go. And if you look at this slide, you can see this slide shows the map of the hepatitis C virus distribution. And we show mostly only those viruses that are known to infect the humans. You can see here, there's a global distributions. And according to the World Health Organizations, each year, more than 20 million people are infected by the hepatitis C virus. And of those, about 3 million cases develop acute viral hepatitis, and about 44,000 of those actually die of hepatitis C virus infection. So clearly, this is an important global public health problems. So for a long time, it has been believed that hepatitis C only infects humans. This is exclusively a human disease. And however, back in 1997, our group discovered the very first animal strain of hepatitis C virus from pigs in the United States. So we named this virus Swan HTV and initially tried to distinguish from the human hepatitis C virus. And now, since our initial discovery, the Swan HTV have now been discovered in pigs essentially from all over the world. And in some cases, such as in the United States, some farms, 80% of pigs are infected by the hepatitis C virus. Now, the infected pig do not have overt clinical disease. And so they are clinically normal, but they do develop microscopic evidence of a hepatitis, as you can see here from the picture on the left. And panel A, you can see here this foci of hepatocyte necrosis. And panel B, you can see here the infiltration of some of the lymphoplasmacytic cells. And so it's typical acute viral hepatitis in infected pigs. So since our initial discovery of the hepatitis C virus from pigs, the virus has now been identified genetically from more than a dozen other animal species. And so these are genetically confirmed and has been isolated from different animal species. Listed here are some of those animal species from which the virus has been genetically identified. And you can see here, there are at least 4 different genotypes, genotype 3, 4, 5, 6, that has been identified from both domestic and wild pigs. And also recently, genotype 7 and 8 hepatitis C virus has been identified from a camel. And you can see here the genotype 3 has been identified from deer, rabbit, and mongoose. And even fish, the cutthroat trout, and also harbor an unrelated hepatitis C virus. And so clearly there's a large animal host for these important human pathogens. So because of the discovery of this large number of animal hepatitis C virus, and the classification of the virus has been changed and actually really evolved almost every year. The virus now is classified in a new family called Hypervariable, and it contains 2 genera. The genus Orthohepativirus, which contains 4 different species, Orthohepativirus A through D, and the genus Pisahepativirus, which contains only the fish Hepativirus, cutthroat trout virus. Now, so far, only the member within the species Orthohepativirus A and Orthohepativirus C are known to infect humans. So if you look at this phylogenetic tree of Orthohepativirus A on the bottom right corner, you see there are at least 8 different genotypes within this species. The genotype 1 and genotype 2 showing here only infect human. They are exclusively human pathogen. There's no evidence that genotype 1 or genotype 2 infect animals. However, if you look at here, the genotype 3, this genotype infects humans, but also a large number of other animal species, domestic and wild pigs, deer, rabbit, mongoose. And similarly, genotype 4 HEV infect human and also pigs. And then there's a genotype 5 and 6, and these 2 genotypes identified only from wild pigs, and genotype 7 and 8 have been identified from camels. And the camel HEV has been shown to actually infect humans. And so clearly there are a large number of animal reservoirs. So one of the study we and many other has been trying to understand is whether or not this animal strain of a hepatitis C virus has the ability to jump species infecting humans. And so I'm just using one of the example here. I'm going to use the Swan hepatitis C virus or Swan HTV. I'm going to show you just a few slides about cross-species infection that we have done in the last few years. And so basically we show that the Swan hepatitis C virus and both the genotype 3 and the genotype 4 can indeed cross the species barrier and infect the non-human primate, rhesus macaques and chimpanzees. And also the genotype 3 and the genotype 4 Swan HTV has been shown to infect humans. And conversely, the genotype 3 and the genotype 4 human HTV, but not genotype 1, genotype 2, can also infect pigs. So clearly there are documented cross-species infection for the Swan hepatitis virus. And just going to show you in a couple of slides. This particular slide, you can see this is a rhesus macaque experimentally inoculated with the genotype 3 Swan HTV. Now, as I mentioned earlier, the genotype 3 has been identified from pigs essentially all over the world. And you can see here, and this rhesus macaque clearly become infected by this pig virus, as evidenced by seroconverting to HEV antibody at about 4 weeks post-inoculation. And there's also a virus shedding in the feces, viremia, between 1 and 5 weeks post-inoculations. And also there's elevation of liver enzyme. You can see here both ICD and ALT at about 4 to 5 weeks post-inoculation. And during the peak liver enzyme elevation, we also see the liver histopathology, which is consistent with acute viral hepatitis. And I should have mentioned here that the chimpanzee inoculated experimentally with the genotype 3 Swan HTV also become infected. So both rhesus macaque and the chimpanzee can be infected by the genotype 3 Swan HTV. And for some reason, this, let's see here, here we go. It doesn't allow me to advance the slides. So conversely, and we also try to determine if the human hepatitis C virus can infect the pigs. So we utilize the genotype 3, which is the US2 strain, and genotype 4, which is a Taiwanese strain of a human HEV. And then you can see here, when we inoculate pig with these 2 human hepatitis C virus, and the inoculated pig quickly become infected, as evidenced by seroconverting to HEV antibody. And we were also able to detect the viremia and the fecal virus shedding. So clearly, this is a 2-way cross-species infection. The genotype 3 and 4 pig HEV to human, and the genotype 3 and 4 human HEV to pigs. So in a very recent study, we want to see if there's no risk for zoonotic infection and also food safety, and because, as I mentioned before, a large number of pigs in the United States are infected by the hepatitis C virus. So we did a really large epidemiological study in which we analyzed more than 5,000 market weight pigs. And those are the pigs that are in the slaughterhouses. And so we obtained those samples from 25 different slaughterhouses in the United States. And we tested for seropositivity. We also tested for RNA positivity. And so if you can just focus on the bottom, the line that highlighted here, because you don't need to look at the other data. And you can see here about 6.3% of those market weight pigs from the slaughterhouses are still viremic. They still have the virus circulating in their blood. So this really raised the concern, because those blood from those slaughterhouses and could potentially contaminate the pork supply chain, and therefore may pose food safety concerns. Indeed, we have shown that about 11% of the commercial pig liver sold in a local grocery store here in Blacksburg, Virginia, are contaminated by the hepatitis C virus. And 11%, that's very significant numbers. And so these are the pork products on the shelves of grocery stores. And you can see here, we sequence all those strains that we recover from the pig liver from the grocery store, and they all belong to the genotype 3. Now, as I mentioned earlier, now you should know that the genotype 3 causes zoonotic disease and can infect the human. So this really poses a concern about food safety, and especially if we consume undercooked or even raw pork products. And so we did a couple of studies try to assess the food safety issue associated with the hepatitis C virus infection and pork products. And this is one of the studies we did. You can see here, we tried to use different cooking condition and to see if we can actually adequately inactivate the contaminated hepatitis C virus from the pork products. And you can see here the negative controls, all the animals are negative, and the positive control, which is the liver homogenate that are known to be positive for HEV and from the grocery store. And they are, when we inoculate the homogenate into the pig rapidly become infected. However, when we stir fry or boil the contaminated pork products for 5 minutes, we can completely inactivate the viruses. However, when we incubate the contaminated pork homogenate at 56 degrees for 1 hour. Now we use the 56 degree for 1 hour to mimic the medium to rare cooking condition in a restaurant. And you can see here that after 56 degrees for 1 hour incubation to mimic the cooking temperature in some restaurant, and we inoculate this homogenate back into the pigs, you can see here the pigs still become infected. So this indicates that the medium to rare cooking temperature in some of the restaurant cannot inactivate the hepatitis C virus from the contaminated animal meat. So if you have, let's see, if you go out for the dinner and order the pork steak and the special medium to rare cooked pork steak, and so there's a chance you may get infected by the hepatitis C virus. In fact, this has occurred many times, and there are a large number of cluster cases and the sporadic cases of hepatitis C virus infection in humans who have consumed raw or undercooked pork products. And I'm going to show you a couple of examples. Showing here is pig liver sausage called fugitello, and this is actually a very good pig sausage from southern France. It's a smoked pig liver sausage, and you can see here 3 families get together and they had a cookout, and so the fugitello sausage is on the menu, and 7 out of those 13 family members who ate the fugitello sausage become infected by the hepatitis C virus. However, none of the 5 individuals from the 3 families who did not eat the sausage had any evidence of infection. But the most importantly, the sequence, the hepatitis C virus sequence recovered from the leftover pig liver sausage is nearly identical to the sequence recovered from the human patient. So indicating the source of the human infection comes from the contaminated smoked pig liver sausage. I'm going to show you another example. And this particular example is a deer meat. And as I mentioned before, there's a large number of animal reservoir, and not just the pig, the pork. And so other animal can also serve as a reservoir. In this particular case, it's the deer. And you can see here 2 families in Japan. They went on a hunting trip, and they caught on a deer, and they decided to consume the raw deer meat, which, as you know, is a delicacy in Japan. And 4 of the patients from these 2 families become ill with the hepatitis E. And about 6 to 7 weeks after they consume the raw deer meat. Once again, if you look at the phylogenetic tree, most importantly, the sequence of the hepatitis C virus recovered from the leftover deer meat is essentially identical to the sequence recovered from the 4 human hepatitis C patients. So this indicates that the source of infection for this 4 human patients come from the deer. So the consumption of the deer meat is the cause of this foodborne zoonotic infections. We also done quite a few epidemiological studies, try to see, determine the zoonotic risk and for infection through the contact with infected animals. In this particular study, you can see here, we analyze the pig veterinarians and pig handlers from 8 different states. And you can see here that the individuals, both the veterinarian and the blood donors actually, from those traditionally major swine states are more likely to be infected by the hepatitis C virus compared to those traditionally not the major swine state. For example, if you look at the state of Minnesota, which is a major swine state, both the swine veterinarians and blood donors are 5 times more likely to be seropositive for the swine hepatitis C virus antibody compared to the individual from the state of Alabama, which is not a swine state. So clearly, occupational contact with pig and other animal species is a significant risk factor for the hepatitis C virus zoonotic infections. In fact, many recent studies have shown that chronic hepatitis C is likely of zoonotic origins. And because the chronic hepatitis C has now become a very important clinical problem. And in those organ transplant recipients, if they infected by the hepatitis C virus, about 60 to 80% of them will become chronically infected. So this is a major concern. And so far, all those chronic HIV infections are almost exclusively found to be caused by the zoonotic genotype 3 and also zoonotic genotype 4. So it is believed that the source of chronic hepatitis C virus infection come from contaminated animal meat. Now, also, I mentioned at the beginning, there are extrahepatic clinical manifestations of hepatitis C virus, such as neurological disease and kidney disease. And those extrahepatic disease are believed to be caused also by zoonotic hepatitis C virus. And about 5.5% of the infected patient actually has neurological disease. And the most common one are the Guillain-Barré syndrome and neurological amyotrophies. But some of them also have kidney disease. And most of the studies so far show that this extrahepatic disease, neurological disease, and the kidney disease, they are linked to both the genotype 3 and the genotype 4 HIV infection, and both of them on a zoonotic genotype. So hopefully by now I have convinced you that hepatitis C virus is a zoonotic pathogen that has a large number of animal reservoirs. And so the next step, of course, is going to be very important for us to understand what are the genetic bases and also the host factor that involve in cross-species infection. And so the lab has been spending the last few years trying to understand both the host factor and the genetic element in the viral genome that may be responsible for cross-species infections. And so I'm gonna just spend just a few minutes today showing you a couple of studies we did in terms of the potential genetic element that determine the virus cross-species infection. If you look at the genomic organization of the hepatitis C virus shown here, they are really simple genomic organization. There are only 3 open reading frames. The ORF1 code for non-structured protein, and it has many functional domains like the methyltransferase domain, the helicase domain, the RNA-dependent RNA replacement domains. And there's the ORF2 code for the capsid protein, and ORF3, which overlap ORF2 code for the small protein that involved in virus replication regulations. So because there's ORF2 is only a structured protein, and our initial focus is ORF2 because this gene code for the capsid protein. Naturally, you would think that the capsid protein would interact with the host cell receptor and therefore determine the host range and determine the cross-species infections. And also we focus on another region called hypervariable region in the open reading form 1, or HVR, stand for hypervariable region. This particular region is very rich in protein residues, and the sequence is highly variable. So those are the 2 regions we initially try to focus on, try to determine if they are responsible for cross-species infection. So just very brief 1 slide data to show you, and yeah, this take two postdocs, I think 4 years to do the study, but we'll just summarize this in one slide. And basically, we take advantage of two very unique reverse genetic system we have. The genotype 1 human HEV infectious clone, which only infect human, and the genotype 3 Swan HTV reverse genetic system. And this virus infect both human and pigs. So by utilizing the genomic backbone of the genotype 1 human HEV and the genotype 3 Swan HEV, we constructed a panel of chimeric virus. We tried to swap the ORF2 capsid gene between these two different genotypes. And not only we swap the ORF2 gene, but we also swap some of the surrounding region, like ORF3 or some of the intergenic region. So we end up with a panel of chimeric virus. We then inoculate this panel of chimeric virus into pigs. And so the idea is that if the ORF2 capsid gene is indeed responsible for cross-species infection, then the chimeric virus that utilize the human HEV backbone but contain the ORF2 from the genotype 3 Swan HEV should be able to infect the pigs. And to our surprise, you can see here when we inoculate this chimeric virus that based on the human HEV backbone, none of them actually infect the pigs, and all of them are still a dead virus when we inoculate into the pigs. Even though you can see here, those chimeric viruses that based on the genotype 3 Swan HTV backbone, they are still infectious when we inoculate into the pig. And we actually further confirmed this study, and we used the genotype 4 human HTV backbone to construct additional chimeric virus, and the results are very similar. So taken together, the study showed that the ORF2 capsid gene or the ORF3 gene are likely not involved in a cross-species infection, which is kind of surprising, and because the ORF2 is the only structural gene. So this led us to focus our research on the hypervariable region, which is in the open reading from 1, as I mentioned earlier. The reason we want to focus on this hypervariable region, or HVR, is because the sequence in this particular region is extremely variable. It differs by as much as 71% between two different genotypes. And even within the same genotype, they differ by 30 to 40% within two different isolates. So because of this extensive genetic variation, the first question we asked was, does this sequence really needed for the virus infection at all? And so to answer that questions, once again, we utilize a reverse genetic system, and we use both the Swan HEV and also we use an avian HEV, a genomic backbone, and we constructed a large number of deletion mutants, in which we deleted the hypervariable region from the viral genome. So I'm going to only show the data here that we use the Swan HEV genotype 3 Swan HEV deletion mutant. And you can see here 3 over 4 deletion mutant. And when we inoculate into the pig, and the pig can still become infected, and they still shed the virus in the feces, they still develop viremias. And one of the mutant, which contain the complete deletion, does not have the virus shedding, does not have the viremia, but this virus does cause seroconversion in the inoculated pig, indicating this mutant is still viable and infectious. So this study clearly shows and that the hypervariable region is dispensable for virus infectivity, and it's not required for the virus to infect the pigs. So the results kind of somewhat expected because the extensive sequence variation within the hypervariable region, but it's also puzzling. And the reason I said puzzling is because if this particular region is not required for virus infection, then why would the virus still keep this particular region in the genome? And the fact the virus keep the hypervariable region still in the viral genome suggests it must play some roles in the virus replication. So we hypothesize that maybe this hypervariable region it's not required for virus infectivity, but it may interact with the host factors and modulate the efficiency of the virus replications. So we first constructed the replicant system in which we replace part of the structure gene of the virus with a luciferase reporter gene. And then we constructed a large panel of deletion mutant. You can see here, we have the deletion of hypervariable region in the N-terminal region. We have a deletion in the middle. And we also have the deletion in the C-terminal region. And so we then tested this deletion mutant in the human liver cell lines. And if I can, okay, here we go. And you can see here the result. And based on the luciferase assay of the promoter activities in the human liver cells, we see that all three deletion regions, the N-terminal, C-terminal, and the central region, drastically reduce the virus replication efficiency. Particularly in the middle central region, you can see here, the effect is very drastic and significantly reduce the virus replication efficiency. So this indicate that this hypervariable region, even though it's dispensable for virus infection, it does interact with host factor and modulate the virus replication efficiencies. And so it indicate it does play a role in virus replications. So at about the same time, we finished up our study and on the hypervariable region, and the C. Emerson's group at the NIH made a really exciting discovery. So they discovered a naturally occurring hepatitis C virus recombinant. And they show that this naturally occurring HEV recombinant has the insertion of the Robinson protein S17 or Robinson protein S19 sequences in the same hypervariable region of the viral genome that we've been studying. And so this is really exciting. You can see here the insertion of S17, S19 into the hypervariable region in this naturally occurring HEV mutant. And most importantly, they show that when they insert this human Robinson protein S17 into the hypervariable region of genotype 1 human HEV, which, as you know, only infect human, this recombinant virus now have expanded the host range, allowing the virus now to infect a large number of cell lines from different animal species, including the pig and the deer. And so this is really exciting. And we thought maybe this Robinson protein S17 have something to do with the expanding the host range. So we decided to look into this. And at the beginning, there's a very limited information about the human Robinson protein S17. So we have to really did quite a lot of work at the beginning to characterize the Robinson protein S17. And so I'm not going to go into those details, but we did a lot of characterization of this S17 sequences. One of the unique features we found is that both Robinson protein S17 and S19, they contain a nuclear localization signal. And you can see here, both S19 and S17 has a nuclear localization signal. So that led us to think that maybe this insertion would allow the virus to have a nuclear stage, and that may explain the enhanced replication expanding the host range. So to answer that questions, and we conducted a number of studies, and show you here is one of them. And you can see here we have two distinct strains of the virus, the P6 showing here that contain this Robinson protein S17, and the P1 virus does not contain this insertion. And so when we express ORF1 of the P6 virus that contain this S17 insertion, and you can see here, the ORF1 protein is expressed predominantly in the nucleus, whereas the P1 virus that does not have the insertion expressed in the cytoplasm. Similarly, when we express the hypervariable region of the P6 virus, which contain this S17 insertion, and you can see here the P6, they're predominantly expressed in the nucleus, whereas the P1 hypervariable region that does not have the insertion expressed predominantly in the cytoplasm. So really this study showed that the ribosome protein S17 insertion actually bestow a new nuclear trafficking capability of the HEV open reading from 1. And so this was kind of exciting at the time because we thought, okay, maybe we'll find the reason why we see this expanding the host range and enhanced replication of this hepatitis C virus with these insertions. And so we decided to say if this nuclear trafficking capability is responsible for the enhanced virus replication and therefore expanding the host range. So we made a large number of mutations within the ribosome protein S17. So we changed the lysine residue to alanine, and we made a single new mutation. You can see here double mutation, triple mutation. We also made multiple mutations. And you can see here that the single mutation of the lysine residue or double mutation did not really change the nuclear localization of the viruses. However, triple mutation or multiple mutation alter the nuclear localization and change the viral antigen expression from nuclear to the cytoplasm. But most importantly, when we look at the replication efficiency of all these mutant viruses, and regardless nuclear localization or cytoplasm localization, all these mutant viruses you can see here have a drastically reduced replication efficiency. And so indicating that these mutants have reduced ability of replication in those human liver cells and also in the baby hamster kidney cells. So the data suggests that it is the lysine residue, not the nuclear localization of open reading from 1 correlate with the enhanced virus replications. And so of course, the next step is to look at whether or not this mutant now have an altered host range and may be able to infect different animal species. So this is something that clearly need to be done in the futures. So, in conclusion, hopefully I convince you that the hepatitis E virus is a zoonotic pathogen. There exists a large number of animal reservoirs, and shown here in this slide. We already know that some of the animal hepatitis C virus, such as those from pig, from rabbit, from camel, deer, and more recently from rat, this animal hepatitis C virus has now been demonstrated to infect the cross-species barrier and infect humans. So these are zoonotic viruses. But if you look at this slide, there's a large number of other animal species. So far, they only has been found and to contain antibody and to the hepatitis C virus. The virus has not been genetically identified from many of the animal species. So clearly, there's a lot of work that needs to be done in order to fully understand the ecology and the natural history of this very important human pathogen. So I'm going to stop here by acknowledging those in the lab who did the work. And over the years, I have quite a number of people working in the lab. I'm not going to name them, but they are the one in the lab who did the works. And also my collaborators, both at the Virginia Tech and other institutions. And our research is primarily funded over the years by the NIH, and we are funded by the NIH since 1999, and so need to acknowledge their financial support. So with that, I don't know if I have time. I'll be happy to answer any question you may have. Thank you.
Yeah, thank you, Dr. Meng, for the wonderful talk. Yes, I think we do have some time, maybe up until 1 o'clock, to entertain some question, if you will, Dr. Meng. So, any question?
Yeah.
There you go.
I have a question, if you don't mind. So, yeah, that's a great presentation. Just wondering, what is the main route of distribution of this virus in an ecological system? Let's say if a wild pig has a virus, then how other species can get it, through what routes, and how likely that other species in the same ecosystem would have it?
Great question, Igor. And the virus is transmitted primarily through fecal oral route and through contaminated water and contaminated food. So in the ecosystem, I would assume most likely through contaminated water, and if not through direct contact. And within the same animal species, most likely through direct contact. But the virus shed a large amount in the fecals materials, in the human stew and animal feces. So those contaminated feces can contaminate irrigation water or even sometimes coast water. There are studies show that some of the seafood are contaminated by the hepatitis virus. So those contaminated irrigation water may serve as a route for infection of other animal species. And but in human, the most common route, zoonotic infection, is through consumption of undercooked animal meat.
But other animals must be susceptible, right? It's not just any animal can get infected, right?
That's correct. And there's more than a dozen animal species harbor the viruses, and so there's a large number of animals susceptible to the virus infections. And now, many of those animal viruses so far do not infect human. As I mentioned earlier, there are only about confirmed 4 or 5 animal hepatitis virus infect humans. So some of them do not infect humans. They may transmit, for example, the avian hepatitis virus from chicken. We know that virus infects turkey, but it does not infect human. So it may transmit among the animal species, but it may not to humans. But there are at least 4 or 5 animal species: a pig, rabbit, a mongoose, deer, and also rat. We know they can infect humans. So I saw there's a raised hand from Andrea Bear.
Yeah, Dr. Bayer.
Hi there.
Hi.
I actually worked with Scott Kenny back at Penn State, who I know who is one of
You did.
So I wasn't surprised to see the nuclear localization work done, because I'm like, I knew Scott was working on that at one point. So my question is there a recognized nuclear localization signal or nuclear export signal within Hep E? And if so, what sort of role might you think that does for the virus in its life cycle?
It's great questions. And yeah, indeed, Scott wasn't a postdoc in my lab for, he's been in the lab for quite some time, but he's not a professor at the Ohio State. And the work I showed actually was done by Scott, actually. And we did not find any nuclear localization signal or even, remotely similar one when we scanned the genome of the hepatitis virus. And the only one we have is that insertion from the Robinson protein S17 or S19. And so actually Scott thought of exactly you thought, and he did the scanning of all 4 different genotypes, could not find anywhere even similarly related to the nuclear localization signal. So the question is, no, we didn't find it. But if we can't, I mean, that would be something though.
Absolutely, thank you.
Yeah.
Any other question?
Yeah, this is Dr. Degwa. That was a great presentation, Dr. Meng.
Thank you.
Yeah, my question is, in your introduction, you showed some data that shows that pregnant women, have a very high case fatality late. So is there an explanation to that?
That's great questions. And we actually received a $2 million last year just to find out why. And so we're getting a new grant from NIH to study what is the underlying mechanism of this high fulminant hepatitis that we see in those pregnant women. And so the answer right now is we do not know. However, there are 3 postdocs in the lab working on the project right now. And we have some promising data. One of the data which we haven't published yet is that Harani, one of the research scientists in the lab, has shown that progesterone actually, and the hormone may actually enhance the virus replication. As you know, both estrogen and progesterone are elevated during pregnancies. And so we do find that those sex hormone actually promote the hepatitis C virus replication. So this is one of the the lead we're following right now, and the data looks very promising. We're getting ready to submit for publication. There are probably other reasons, immune regulation. As you know, in pregnancy, there are a lot of immune downregulations, and that may also contribute to the severe hepatitis. But the answer is we do not know exactly, but looks like at least the progesterone is involved.
Oh, great. The other question is, you've done quite some work with this virus. Do we have a vaccine yet that we can use maybe from your deletion mutants or something, or is it there?
Yeah, there is no vaccine available now, and unfortunately, the vaccine is only licensed for use in China now. The group in China developed a vaccine, and the papers published in Lancet. The vaccine has been really effective, and the reason why we stopped developing the vaccine, our group, is that there's really no market, and because many of the industry vaccines, they do not want to support it, and because this basically only seen in developing countries, and it's very difficult for us to get funding and to do the vaccine study. And so we do have a recombinant capsid-based vaccine. We test the vaccine in pigs, and it shows actually very effective. But we never done any human clinical trial, which is something I would love to do, but we just doesn't have the resources to do that. And so the vaccine is available in China. I think recently they are doing a trial in another country in Southeast Asia in pregnant women, actually. So hopefully that vaccine will be used in other countries also.
Oh, great. Last question. I work with food animals, and in pigs, what do you think is the main route of acquisition of the virus? Because most of these pigs are reared indoors. And
yeah, they are fecal oral, and because a large amount of virus is shed in a pig feces, those pigs, they sniff around, and so the fecal materials that contaminate the feed and contaminate water is the one that transmits. So fecal oral route of transmission for the pigs.
All right, thank you so much.
Sure.
Dr. Meng, I have two questions, even though I realize it's over 1 o'clock now, but hopefully brief questions. One is that, have you tried to look at whether how often recombination happens in the different strains for different genotypes of?
We did not. Yeah, we did not. But there are quite a few studies, and most of the recombination that reported so far are actually reporting some of the human sequence actually and inserted into the viruses. So kind of strange, and we already saw the Robertson protein S17, S19, but there are some other human sequences inserted into the viral genome. And there are, I think, 1 or 2 studies, they show intergenotypic recombination, but those are really not independently confirmed yet.
Okay. And the other thing is that you have alluded to this during your talk. So here you look at the how the different genotypes of virus differ from each other. I was wondering whether the receptors on the human cell or potential host cell, the difference among the receptors could also play a role in terms of how different genotypes could.
Great question. And I would assume there will be difference, but unfortunately, the receptor for the hepatitis C virus have not yet been identified. So we don't know the HEV specific receptor yet. Several group has tried for many years. Our lab, one of the former postdoc tried, I think 3 or 4 years. No one so far has been able to identify the cellular receptors, specific cellular receptor for HEV. And this is one of the area that is a huge gap in the research area. And but we do not know the receptor yet.
Okay.
Yeah.
All right. Hopefully we will know sometime soon.
Yeah, well, hopefully. Yeah.
Okay, great. With that, I think, again, it's 1:02 now. So I guess we'll finish our seminar here. Let's give another round of applause if you can clap, you're welcome to unmute yourself and clap for a great presentation.
Well, thank you. It's a pleasure. So, thanks. Take care. Yeah.
It's
Thank
great,
you.
yeah, great honor and a pleasure to have you here. All
Yeah,
right.
thanks.
We'll see everybody next Friday. All right, for another interesting seminar. Until then, take care. I'll see you next Friday. Bye now.
Bye.